Line now. Hi everyone, thank you for coming, even those that walked through the rain. But now you got to see the beautiful new science faculty building. So it is a real great pleasure to have Alessandro here, Alessandro Vannini. This is not also Alessandro Borsellino, who used to be my student, who's now in his lab. So I don't know how you guys are dealing with that, because some maybe you have nicknames for each other, but we can hear later.
It's complicated.
It must be. All right. So Alessandro is from Italy, so he studied in Rome. Well, it's really great. He studied in Rome, and then he did, after his PhD, he went to the lab of Patrick Kramer in Munich, was it at the time?
Yeah.
Where he did his postdoc, where he first got introduced to RNA polymerases. Then, if I got the numbers right, in 2012, he actually moved to England, to the Institute of Cancer Research, where he set up his own group, continued working on RNA polymerases. And this is where I actually first met him. I organized a one-day meeting on protein crystallography. For some of you, that is an old technique to solve structures. It still is around, but it's getting very rare. So this was for the English Crystallography Society, and I organized a meeting, and I invited Alessandro. And he said, yeah, I work on Pol III, and I freaked out because I also worked on Pol III, but I worked on DNA Pol III, and he worked on RNA Pol III. So it was okay, it worked out well. Anyway, since then, he's been doing marvelous work, and I also learned that there are 3 RNA polymerases. One of them is making, you can correct me later. So RNA polymerase 1 is mostly making ribosomal RNA. RNA polymerase 2 is doing all the mRNA, and RNA polymerase 3 is the one that's doing all the non-coding RNA, which at the time maybe looked really suspicious and weird, but now we know they have so many functions that we don't fully understand. So maybe it's actually the most interesting RNA polymerase to work on.
Absolutely.
So, there you go. I thought I had that talk to you.
Thank you.
So in 2019, he moved back to Italy, to Milan, where he now is the head of structural biology at the Human Technopole, which is a brand new building, which is still being built, if I understand correctly, where he was incentive to set up the whole structural biology unit with lots of cryosis and glacials, and everything is working marvelous. Right?
Absolutely.
And there he's continued his really great work on RNA polymerase, but also looking at other enzymes involved in DNA, in this case, in DNA condensation that we could condense in. And hearing tidbits from my former student, Alessandro, it's actually going really well. And I think you got something coming out soon that you may or may not talk about now. So the floor is yours. Great for having you here.
Thank you.
As I said, I'll be sitting in the back because I have to sneak
up
at the end. But
thank you. Thank you very much for the very kind invitation. I'm in the right field. All right, good. Yeah, thank you for the nice invitation and for the nice introduction. It's true, yeah, me and mine that met like a decade ago or more back in Cambridge, and we work both on polymerases, but different type of, luckily. And so yeah, as Minder said, I'm mainly interested in transcription, as you might know. This is what is referred to the central dogma of molecular biology, and this is really like let's say a figure taken from one of the famous Francis Crick's lecture at UCL in London, which is annotated scheme where obviously the genetic information contained into DNA is translated into protein passing through an RNA intermediate. This process is called gene transcription, and in eukaryotes, the nuclear genome, or excluding the mitochondrial DNA, is actually transcribed by 3 distinct RNA polymerases: Pol I, Pol II, and Pol III. As Mandert already pointed out, Pol III is the most interesting one. I'm just kidding, but Pol I is really a dedicated machinery. It just transcribes one single gene, despite we have many copies of the largest, so the 45S ribosomal RNA. This is the largest ribosomal RNA subunit, and it just transcribes that gene. So it's a very specialized machinery. Pol II transcribes all the mRNAs, microRNAs, some non-coding RNAs, long non-coding RNAs are also transcribed by Pol II. Pol III is again more eclectic, so it transcribes the entire pool of tRNAs. The 5S ribosomal RNA, this is largely overlooked, I think, in the field of ribosomes, etc., or ribosomes regulation, etc. It undergoes a lot of regulation, as much as the largest subunit. but also the small nuclear RNA, for example, this is harbors the catalytic side of the spliceosome, for example, and another, this is part of the signal recognition particle. So all very short, essential, non-translated RNAs. Usually the common feature is that they are very rich in secondary structure, and they are shorter than 500. I think the longest one is 500 base pairs, so really short. So we are interested in that because obviously deregulation of this machinery give rise to severe diseases. Once Pol III, well, all the polymerases were, well, apart from Pol II, were supposed to be like housekeeping machineries that just produce a lot of tRNAs. This is actually really far from the truth. There is a lot of regulation that underpins the activity of this. And so it's clear that Pol III, for example, is one of the major effectors of aging in a torque-dependent manner. It's a downstream effector of torque. It's obviously involved in cancer, so the regulation or upregulated Pol III transcription is observed in all forms of cancer, I would say, in almost all forms of cancer, and we contributed describing a subset of genes which are really important. I'll talk briefly about that. It seems that tRNA biology is also very important as it's connected to very important neurological disorders. I'll also mention something, and this is quite new. And it's a field that we are really also trying to get into very slowly. This is very important as Pol III seems to is the only one of the 3 eukaryotic polymerase that has an activity in the cytoplasm. And what does it do in the cytoplasm? It detects cytosolic DNA that can be leaked from the nucleus, or importantly, can sense viral DNA. So when DNA based viruses enter the cells, they might leak a little bit of their nucleic acids, which I don't believe much, but maybe in their cycles there are phases when they bud from the nucleus back in the cytoplasm where some of their and this is where Pol III detects them and actually elicit a potent immune response. And this is very important. It's really a novel field, and we are looking into that. So the lab is basically, in a way, divided into 2 fields. One is the more canonical, mechanism of Pol III transcription, how it's regulated, how gene transcription works, both, in yeast, in human, in higher eukaryotes, and how this really, the regulation contributes to disease. And there is a, let's say, novel, so we started already a couple of years ago to look at how Pol III transcriptional unit contributes to the structural integrity or structural organization and function of the genome. So those are called extra-transcriptional functions because they do not depend on the Pol III machinery's activity or the capacity to synthesize specific RNA, but rather Pol III genes or Pol III specific transcription factors seems to be really involved into several functions. As heterochromatin barrier, they are enriched at tag boundaries. They are important for stalling gene looping and SMC complexes. They represent a replicative block, and that's how we entered the field that Minded already mentioned, which is the one of SMC complexes, so structural maintenance of chromosomes, so cohesin and mainly condensing complexes. So what I will do today, I will go through few, let's say, important discoveries that happened in my lab, but I will, and this will, of course, be in the field of like more canonical transcriptional role of Pol III, in particular an unpublished story regarding transcription of small nuclear RNAs. So all the components of the spliceosome, they are transcribed by a mixture of Pol II and Pol III using a common set of transcription factors. And this is really a let's say, a very exemplary case of a transcription factor that is shared among two polymerases. It's really an exception, sorry. And then I'll also talk about extra-transcriptional functions. I won't mention this, but this was a very interesting story where we showed how in yeast, a specific set of retrotransposons use their integrase to specifically recognize Pol III genes and integrate specifically few base pairs upstream of Pol III genes. And this is because Pol III genes have coding regions which are, well, promoter regions which are within the coding region. So having an insertion upstream doesn't cause any problem to gene expression. But the field where we are more actively working on is the condensing field, where we already had a couple of, let's say, publications and initial results. These were negative staining 2Ds back in 2020, and Alessandro Borsellini, which some of you know, really kind of inherited this project and really pushed it, and really now has a beautiful story that I will talk about. Hopefully, it will stay unpublished for not too long. I hope, I would be happy if by the end of the year this will will come to light. But and then, as I said before, this is also something that it's a little bit premature to talk about, but I just want to mention that we are using an in situ approach to look at how Pol III recognize cytosolic DNA. So we are using, this is actually expansion microscopy, where you cross-link your material in a sort of a nappy for kids, just a gel that you can then expand physically 5 or 10 times, and then by immunofluorescence you're visualizing your proteins of interest or complexes of interest. This is just a perinuclear cytoplasmic DNA. This was just transfection with lipofectamine, but shows already that some nucleic, so Pol III really accumulates in distinct foci along these kind of vesicles, which is already a good kind of preliminary result that shows that this is quite specific, forming these kind of factories. We are also working with a real virus, so HSV non-replicative, and we have really interesting results of accumulation of Pol III in regions which are around the endoplasmic reticulum, etc. And so we are now pushing this to visualize these things in situ with a correlative approach. But that's very, very early days, so I don't feel comfortable yet to show it. All right, so let's go to the real deal of the business. So this is a schematic of how Pol III promoters are organized. As I mentioned before, the control region, the promoters themselves, are typically inside the coding regions. There are just very few exceptions, and I will talk about this. This is more canonical upstream promoters, similar to Pol II, for example. And those are instead all tRNAs and ribosomal RNA really rely on sequences which are decoding within the coding region. Also, for activity, Pol III doesn't really work by itself, but really relies on a set of auxiliary transcription factors. I don't want you to remind all these names, but just the fact that the minimal activity is conferred by what is called the pre-initiation complex, which is Pol III plus this trimeric TFIIIB complex. This is capable of, as long as you have a Tata box positioned correctly, this initiate transcription faithfully at the right time with full efficiency. So this is the minimum set of what you need for activation of Pol III in yeast. So the other factors, TF3C, SNAP-C, are just recruiting factors for TF3B, which in turn recruit Pol III. And so initially we concentrated on this bit, for that reason, on this bit of what is a full pre-initiation complex. And initiation is one of the steps that is majorly regulated. And so we did, we were crystallographers, but during my postdoc I started already to work on EM approaches. And actually this, I think it's a micrograph that comes from here, if I'm not mistaken. So the first 300 kb data set that we collected for the minimum Pol III pre-initiation complex was collected here around 2017, 18. And yeah, this gave beautiful 2D classes, beautiful 3D reconstruction of the entire thing. This is again back in 2018. And this really allowed us to explain why TF3B is needed for this activation. First of all, recruits Pol III at the right side, at the right genes, but also activates with this protein B double prime and part of BRF1 the polymerase. So this protein, B double prime in orange, really stabilize somehow two subunits of Pol III that in the apostructure are really disordered in a conformation that is capable of opening the DNA. Opening the DNA is the first thing that has to happen when you have to transcribe a gene. So the template strand has to go into the active site, and to do that, you have to open it. This happens spontaneously in the case of Pol III. You don't need helicase activity. You don't need ATP hydrolysis. It's just the energy of binding and these allosteric changes that are mediated by this protein that make it happen. And yes, that is required for full expansion of the bubble in the template strand. Is a specific domain of this trimeric transcription factor. We then moved into so we purified Pol III from yeast. Yeast, as long as you have a fermenter or a lot of flask, you can, produce 200 liters of it, and it's not very abundant. And then we said, like, okay, why don't we do it in human cells just to make our life really miserable? I mean, not mine, to be honest, but the postdoc that was working on it. And so we engineered in collaboration with the Christoph Engel's lab in Regensburg to CRISPR-Cas9, tagged subunit of Pol III. So this is endogenously tagged, where we could grow 80, 90 liters of HeLa cells per prep and get, like, I don't know, 20, 30 micrograms of Pol III, which was very pure, at least. And then we actually, I'm not showing it here, we switched to HEC293 cells, and we get about 10 times more. So, if you want to look for an endogenous complex, in general, don't go for HeLa cells, go for HEC293. They are better, they are more, I don't know, juicy in terms of, at least in our case. And so this was interesting that we solved the apostructure of Pol III, which really is very, very similar to yeast. However, what was very interesting was the possibility to start to map important mutations that occur on Pol III. And those are two, let's say, largely two types of mutation. One that give rise to neurological disorder, even though with very different diseases and different let's say, phenotypes, and the common feature of these mutations that are all mapping at the interfaces of subunits. They were all loss of function or mutations that really destabilized the enzyme. So it was clear that there is something that having less productive Pol III transcription, probably tRNA synthesis, give rise to severe neurological disorders. Why does this happen just in the central nervous system versus other tissues is not entirely clear. Could also possible that you have cytosolic stress, in the sense that these mutations can probably give rise to aggregates that are not really well tolerated. Interestingly, there are another set of mutations which are linked to viral hypersensitivity, and this is the secondary function of Pol III in the cytoplasm. So those patients which are much more peripheric and in region of the polymerase that just bind to double-strand DNA are really hypersensitive towards, for example, the varicella zoster virus, right? So this is chickenpox. So it's a virus that most of us deal with pretty well. Those patients are actually, for them, is actually really, for some of them is fatal. For them, they get reinfected. They have severe let's say symptoms from this. And this makes sense with the more peripheric sort of DNA binding path, because it probably has to do with the capacity of Pol III just to bind DNA or recognize DNA in the cytosol, while in the nuclear polymerase is probably going to be unaffected. And then I go back in time and go back to the time of crystallography, around the time when we met with Mindert, because there was one of the milestones really of my lab was this serendipitous discovery where we were looking at this version BRF2 of this protein, part of this TF3B complex, which is really like, again, it really is important for the transcription of a very, very small subset of genes. The bulk of Pol III transcription is dependent on another protein, which is called BRF1. It's highly related, but BRF2 is overexpressed and amplified in many forms of cancer. And you see here this dependency on high expression levels means really that this type of cancer, in this case breast cancer, are really addicted to have high expression of this protein. The question was really why is that, and why do you need in evolution the emergence of a transcription factor for Pol III that really controls 6 genes? I mean, it didn't make any sense. One of which is the selenocysteine tRNA. And what we discovered, this is a crystal structure, a beautiful crystal structure, a lot of biochemistry that could be explained. But really, the main finding was that there was a small structured element, a 310 helix, with a cysteine at the very tip of it, which really pin together this entire complex, right? So we called it the molecular pin, because if you mutate this to a bulky residue, basically this complex is not stable anymore. And it's like, okay, this is important. And then the next question is like, why do you need a cysteine there? A serine or even an alanine could do the same job. And then the realization was that this is very similar to what happens in an NF-kappa B transcription factor. And long story short, this is basically redox regulated. It confers the cell the capacity to regulate what we think is the selenocysteine tRNA, another BRF2 dependent transcript, to be regulated in a redox dependent manner. So in a normal cell, when the oxidative stress is too high, or you have too much oxidative stress, what happens is that the cells turn off selenocysteine tRNA production throughout this BRF2 regulation. And this give rise to truncated selenoproteins, and this is like a pro-apoptotic message, so that brings or pushes cells into apoptosis, normal. Now in cancer cells, they really thrive in oxidative stress condition. Actually, reactive oxygen species are used as messengers, and in cancer cells you have a very strong, let's say, resistance towards these species. So the idea is that really you need high levels of BRF2 to really keep sustaining the synthesis of selenocysteine tRNA. So you still have selenoproteins. Selenoproteins are mainly either detoxification enzymes or enzymes that are involved in biosynthesis of selenocysteine per se. So it's actually a self-sustaining feedback loop. And we did some experiment in living cells. I cannot say in vivo because otherwise my colleagues that use mice or even higher mammals will just look at me really badly. So in living cells to really support this. And now, of course, we are even more interested in it. I'm going towards an unpublished story, because these type 3 promoters, these BRF2 dependent promoters, are very, very interesting. So first of all, they have the architecture. They are very often, actually in all cases, linked to a neighboring Pol II gene. The neighboring Pol II gene are also somewhat involved into, let's say, oxidative stress or DNA damage. And so the idea is that there is an enlarged BRF2 transcriptional program that can be regulated in a redox-dependent manner. And the other interesting thing is that they depend on this upstream factor, which is called SNAP-C, and a very well-positioned nucleosome, which is brought there by this OCT1 pioneer transcription factor. So again, we don't know what exactly positioned this nucleosome, so we are working. So we have now a structure of the nucleosome with the OCT1 in order to try to create like a sort of a Frankenstein atomic model of this thing. I'm not talking about this today, but I'm going to talk about this today. And so what is also interesting is that I say, one of the genes regulated in this way is the U6 spliceosomal RNA. All the other spliceosomal RNA are transcribed by Pol II, which uses a very similar architecture. And this is, as I said, an exception. Usually they're very divergent in terms of like promoter architecture. It uses the same multi-subunit factor here, SNAP-C, but a canonical TF2B factor. You also have a position nucleosome here. And what really makes one transcribed by Pol III versus Pol II is the presence of a Tata box. So if you have an upstream Tata box, this becomes Pol III transcribed. If you ablate this strong Tata box, it becomes Pol II. So while we know that when TF2B is there, then it's transcribed by Pol II, and vice versa, we don't know how it's possible that this transcription factor really recognizes two different transcription factors and then recruits the right polymerase for the job. And so back in the days, we started to work on this, and in collaboration, oh, sorry, that shouldn't have happened. That's really bad resolution. With Patrick, because he's the Pol II master. And so we provided all the upstream transcription factors, NAPC, as we were working on this. And we obtained a first reconstruction of a Pol II SNAP-C pre-initiation complex. We'll see later. And we started to work on this part. And so this works well. So this is a lot of work because, as I said, human RNA Pol III, you don't get much of it. Then you have to express all these other factors. We usually do glycerol gradients and cross-linking with homeopathic glutaraldehyde concentration, etc. And you always end up, if you want, I can show you a picture later. You always end up using fractions that they don't even enter the gel. You have to. So we used actually mass spec and mass photometry to look at what are the fractions which are likely to contain the entire thing. Because at the beginning we were imaging the wrong things, which when you look at just one image, it's like, oh yeah, you see particles and they look kind of homogeneous, that's great. No, so just like this made difference. And also optimization of the amount of cross-linker, that made a huge difference. And you couldn't tell just by looking at the gel. You need to use something else. But we can discuss this later. And so we typically correct massive data sets. This is 1.3 million particles to end up with, I don't know, 11,000 particles. That gives you like a 3.4 or 3.2. And this is still very annoying to me. Partially is because of the biochemistry. We know that full-length SNAP-C is auto-inhibited somehow, so very few complexes give rise to an active transcriptional complex, and we know now why. But there is also something inherent with, vitrification and cross-linking, etc. And I think it's still kind of really bothering that you start with a million, so you end up with 1% or 1 per thousand of the particles in your final reconstruction. However, whatever it works. And so again, what we obtained was three different states, kind of similar, but with differences. I'm gonna just talk about this. And so what was the discovery that also in this case, basically we gave the polymerase a double-stranded, completely annealed DNA. The polymerase was capable of opening and loading the template strand in an ATP-independent manner. This was not known for this type of promoters. You can see here from the interactions with the transcription factors that basically all the DNA is almost encapsulated, and you have really strong interactions with the upstream part. And what was surprising, though, is that in this case, you don't need that activation that depends on B double prime. B double prime is quite conserved. It's the orange protein that I talked about it before in yeast, that you needed in yeast to really stabilize the conformation of the polymerase that is capable of opening the DNA. It seems that the human polymerase, because it has actually a conserved iron sulfur cluster around this area, is already per se sort of like pre-activated in a conformation that is capable of opening the DNA strand here, or to stabilize the open form. And this was relatively surprising. Now we are finalizing the experiment to show that there is inherently a difference between yeast and Pol III. In yeast, you need this B double prime dependent activation. In human, this is dispensable, and B double prime is just part of this trimeric. It's a recruiting factor again, without having to activate anything. What was very interesting is now when we compare the Pol II SNAP-C complex with the Pol III SNAP-C complex, right? So those are sort of superimposed on TBP, which is a common let's say, transcription factors that induce this bending of DNA as an anchoring point. And you see that the architecture of the two is radically different. Even the DNA path is completely different. Despite these multi-subunit transcription factors, SNAP-C is absolutely the same. So this already points out towards the fact that there are completely different way of recognizing two distinct transcription factors. What you see here, you have these two winged, what we called wing 1 and wing 2, sort of a butterfly open structure, while in this case the wing 2 is disordered, we can't see it, and the wing 1 is interacting in this part. And so if you now superimpose this wing 1, which is the main interactor in one case with the BRF2 and the TF3B Pol III transcription factor versus TF2A, which is the equivalent, let's say, in the Pol II case, you see that basically it uses absolutely the same interface to interact in a very different manner with two completely unrelated transcription factors, one of which select for Pol II and one of which select for Pol III. And this is, again, one of these moments of realization of where you see how evolution has driven these proteins also, or protein interfaces, to perform distinct functions or distinct molecular recognition, right? So we always tend to think about specificity, which is the case, but on the other hand, one has to recognize that sometimes is more complex than just look at the specific interface. And what you also see here, which is important, if you superimpose, let's say, the Pol II conformation on SNAP-C by using TBP into the Pol III transcription factor, you see that there is a massive clash of the wing 2 with BRF2, right? So this, in fact, and this explains why we don't see this part in the electron density, and also explain why when you have BRF2, this really selects for this conformation and then ultimately position the polymerase in the right way to perform to initiate transcription in a faithful manner. And so this really explains now why when you have BRF2 there, you recruit Pol III, and there is no way that this can go wrong and recruit Pol II. And likewise, when you don't have a Tata box and then you have TBP and TF2A that recognize maybe Tata-like sequences but not a strong Tata, in that case, that is going to become a Pol II because there is no way that would let accommodate Pol III, which is, I think, a very nice case to look at how evolution has gone to create these specified systems. Another important feature was to look at that cysteine residue in BRF2, which back then in 2015 was, in the context of a crystal structure of a trimeric complex, and the question was whether that cysteine would still be kind of exposed to be regulated in a redox manner, and this seems to be the case. It's not entirely buried. I mean, it's of course partially protected, but there are no other additional subunits that really cover access to that and reinforce our model of the cysteine as a regulated one. And that's where I switch to the next story, which has to do, as I said before, with the more extra-transcriptional role of Pol III or Pol III units. Actually, I'm going to talk about condensing, which is not really per se, a Pol III factor, but I will tell you why we ended up studying there. They are not completely unrelated. And so this is just to remind you that especially in the human genome or primate genome, not necessarily mammals genome, Pol III loci really represent a good part of our genome. So it's estimated to be 15–20%. And this is basically not because of the Pol III genes, which are relatively abundant but not so abundant, but because of the expansion of retro elements mainly. So sine transposon, so ALU elements or B elements in mice, for example, represent, a good part of our genome. And those are based originally on Pol III transcriptional units. This was, a degenerated sort of based on a tRNA in case of allo elements that has been used as a promoter for these retro elements. I mean, of course, not all of them, actually very few of them are active. They can be activated in form of stress, but the important thing is that you see occupancy of these transcription factors at this element, and many of these elements. And there are also extra TF3C binding sites, which are devoid of a specific coding, a specific promoter region that really recruits promoter. Here you don't see any of the Pol III machinery, but you still have quite a lot of sites. We don't know what is the function of that. Those are conserved in mice. You start to see them already in yeast, this extra TF3C binding site, which are specific. So there is a specificity. It's not just random cheap sec data, and we don't know the function of it. And how we ended up in the condensing field was the realization that very often there is a co-localization of SMC complexes with TF3C. So this is TF3C where there is also Pol III, transcribing Pol III. You see this strong co-localization with condensing 2. This is cohesin, for example, but also with these extra transcriptional sites, right? Of course, co-localization doesn't necessarily mean there is, functional relationship. However, there were some initial experiments in yeast where you can basically, you see you have mutants of the condensing complexes. We don't segregate the chromosomes well; they get stuck, and those are temperature sensitive mutants that you can rescue by using a TF3C mutation. So if you make TF3C less stable, you can totally rescue this, which kind of suggests that there is actually a functional relationship between the two. And so we started to look into this, and then I realized back then that we don't really know how these SMC complexes really work. There was quite some work done in yeast, but not in the human system. And so we used what we were really good at, which is reconstituting recombinant complexes from insect or mammalian cells. So we can reconstitute both condensing 1 and 2 in humans or in metazoans. You have two condensing complexes. In yeast, you have just one. And so we could demonstrate, biophysical characteristic. For example, nucleosomes are good substrates, but they're not really barriers. So condensins can just go through chromatinized template pretty easy. And we started to look at the structure and the architecture of those. This was around 2020. So my lab was still in London, and then there was, COVID happened, and moving the lab, that wasn't easy. But we could already obtain this was based on negative staining. And I remember when we started this project around 2017-18, there was still the notion whether we could visualize in cryo-EM a single coiled helix, right? Is it possible? Maybe not. I mean, why are you going this? And I say, I don't see why it shouldn't be possible. It's about, if you can visualize, or if you can identify this particles, but then it should be doable. And of course, this proved to be correct. And so that is where Alessandro took the project. Alessandro Borsellino took the project on, and we ended up looking at specifically at condensing 2, because condensing 2 is really the initiator of a very important process that happens just before mitosis. So every time a cell divides, which is the initiator of what is called chromosome compaction or condensation before mitosis. So the X-shaped chromosomes that we all know depend on the activity initially of condensing 2 and then condensing 1, which grow shorter and shorter loops, condensing this. However, there is one major problem in all of this, that condensing 2 is present in the nucleus throughout the cell cycle, in interphase, etc. So how does that work? Why this does happen only in mitosis? And so, long story short, we discovered that there is a protein which interacts strongly with condensing 2. And these, the results that I'm going to show are in collaboration. Some of the results are from Andrea Musacchio and Ben Rowland lab, which is actually in Amsterdam at the Cancer Institute, which serendipitously also found the same, or coming from different backgrounds. Andrea was looking at centromeric protein and found condensin 2. We were looking at condensin 2 and find the centromeric protein. Ben was looking at regulators of condensin 1 and found this as a potential regulator of condensin 2. And so this is, just for you to know, this is a very central protein for centromeric identity. This was well known, is part of a larger complex. This is necessary for the correct deposition of SAMP-A nucleosomes, which define what a centromere is. So this is bona fide, only known in the field as a centromeric protein which is essential for establishing a centromere. And so what we discovered was the strong interaction, and this was validated by pull downs, etc. So of course we use AlphaFold, and AlphaFold really could predict this this interaction. Actually, there are also other regions that are probably interacting, but this is the main determinant of the interaction. These 5 residues, and Ale pushed the EM to a decent level. This is, I would say, intermediate resolution, but the quality of the map is actually much. I don't know why Ale always ends up with maps that look better than what the nominal resolution is, contrary to many others. But with AlphaFold prediction, you can really feed confidently models, and we see this extra density of M18 BP1 likely exactly where the AlphaFold predicts this to be. And importantly now, if we mutate this motif into alanine residues, what you see here is the disappearance. So here you see this double band here. This is where NeCEN BP1 fragment co-elude in gel filtration with condensin 2, so quite a strong interaction. And now when you just mutate these 5 residues, right, into alanine, this is basically lost, and all the M18 BP1 is eluding alone, so they do not interact anymore. What we were really pleased and in a way shocked is that if you now use this mutant, so by which M18 BP1 is still perfectly fine at defining what the centromere is, so its function at defining, in centromeric identity is untouched. We just mutated these 5 residues that are mediating the interaction with condensin 2. You still see SAMP-A as it should be, so centromeric chromatin is fine, but you basically don't load and you don't recruit condensin 2 anymore at all in cells, in mitotic cells. And even more importantly, if you look at the capacity of these cells to really compact chromosomes at mitosis, when you use this M18 BP1 mutant, in this case we mutated, I don't know why, it was 5 residues. Originally we thought about mutating 4. You see this massive increase in condensation defects that basically almost mimic a complete KO of condensin 2. Usually when you knock out condensin 2 or condensin 1, the cells kind of survive because rely on the activity of the other. I mean, they're not doing really well. They go on for a couple of cell cycles and then eventually they die. And that's how Ben Rowland found M18 BP1. He was using an haploid cell lines with a knockout of condensin 1 and looked for factors that become essential only when you knock out condensin 1 and identified M18 BP1. So that is quite important. And so what is even more interesting is that then if you look at this motif, it's really similar to a motif of a protein which is called MCPH1, which we worked in the past, because this is a negative regulator of condensin 2. So when this binds to condensin 2 cannot bind very well to chromatin. It somehow, by mechanism which we don't know yet, promotes the turnover of condensin 1. And of course, we did a AlphaFold prediction of this, and look at this. Those binding sites is basically the same, right? And so the idea is like, okay, these two must compete for the same, these 2 factors, one negative and one positive, albeit using a very similar binding strategy, must compete for this. And so what we realize is that this binding site have bona fide CDK1 phosphorylation sites. And I say, oh, wow, this could be the molecular switch that actually prevents MCPH1 to negatively regulate condensing 2 at the onset of mitosis. And so this is just fluorescence anisotropy experiments where you can see that phosphorylation of, in this case, M18BP1, basically at this site provide an additional interaction, so that the basically increasing the affinity for M18BP1 when it's phosphorylated, while at the same time CDK1 also promotes a phosphorylation event of MCPH1 with on the serine residues that actually decrease its affinity. So it's basically switching one from another. It's not a complete on-off thing, but it's just altering this equilibrium. And I'm not showing this here for the sake of brevity, etc. But what we discovered is that really, so what is more important? MCP. So how does that work? So basically, M18BP1 is required and necessary to activate condensing 2, because if you take a cell where you knock out MCPH1, you still need M18BP1 to load condensing 2 on chromatin. So there is another mechanism by which M18BP1 not only binds there and displays. So it's the other way around. It's MCPH1 is basically important for not having condensation during interphase. So M18BP1 condensing 2 is almost an obligate complex necessary for loading condensing 2, for activity of condensing 2 on chromatin, and you need MCPH1 during interphase to sort of displace part of that M18BP1 protein and to alter that equilibrium to a level where you don't start condensate prematurely. And this is quite important because it's a complete novel factor that was, as a moonlighting function in this, while it was supposed to be just a centromeric protein. Now there was a very recent Cell paper that showed that actually the centromere in human cells, it's a bipartite structure, very beautiful, and depend, guess what, on the presence of condensing 2 to hold these two things together. And they don't know what is the factor that brings condensing 2 there. And so, well, I think we know what it is, and it's probably M18BP1 that does these 2 things. So the 2 functions are separated functionally. However, there is this connection between centromeres and condensation, which is very important. And probably it's also where the condensation process starts, where you have an enrichment of condensing 2 at centromeres, and then it spreads towards the arms. And so this is a simple model recapitulating what I've said. In interphase, you have prevalently MCPH1 binding, which limits the binding of M18BP1. So condensing 2 is partially deactivated. So, the genome is not completely unfolded, but it's not condensed as it is in mitosis. And then because of the activation by CDK1, MCPH1 is basically displaced or outcompeted by M18BP1 that comes in and starts this massive cascade of condensation. And then after nuclear breakdown, you have also condensing 1 coming in and and finish off the job. And so what I'm going to show you here is just, which is this last slide that I prepared like 3 seconds ago. We are using also optical traps to look at this on a basically on a C trap. It's a Lumix. It's a confocal microscope combined with an optical trap, where you can basically trap a piece of DNA. In this case, we have incubated just with condensing 2, and then you are going to check what happened to the DNA by stretching the DNA afterward. And so if there is condensation events or loop extrusion forming, you would see that you need less tension. So you could stretch the DNA less, basically. So here, nothing really much happens. Condensing 2 is also a pretty lousy loop extrusion machinery, and in absence of ATP, nothing really much happens. What you see already here, just by this is pre-incubated with condensing 2 and M18BP1, what you see here already, you should, when it takes a while, I should have. There are many more molecules loaded. So this condensing 2 is specifically labeled with quantum dots, I think, on a strap tag. Well, yeah, you can't really appreciate it, but you have to trust me, there are more molecules. And you cannot, you will see that you can stretch this less than with the condensing 2 only sample. And what you will see here is a massive difference. So now when you put condensing 2, M18BP1 and ATP, right? So you will see what this means. So again, we don't exactly know what happens here. We just assume that it's the loop extrusion process going on. But now when you start, when we start to stretch the beads back, hopefully soon. Yeah, here we go. First of all, you see that here, yeah, and that's repeated for many measurements, etc. So obviously, you really require M18BP1 is not just a loading factor, but it's really required to activate somehow condensing 2. This is kind of preliminary, but we think that combining this with cryo-EM, where Alessandro really, I didn't talk about it much because again, it's a little bit premature, but now he figured really out how to deal with this compost. The structure that I showed, and I didn't talk much about it, is at 7 angstrom. It reveals probably also, this is probably an inhibited state, an out-inhibited state, and we believe that M18BP1 is capable of releasing that out-inhibitory state only when in presence of DNA. This structure, only when in presence of ATP, sorry. This structure was in absence of ATP, so the ATPase domain are not engaged, so you don't see this full activation in this structure. But Ale now got a very beautiful below 4 angstrom structure of APO condensing 2 with ATP. So no M18BP1, and this is in a conformation that would not be able to accommodate DNA. This also is a structure that they got with DNA. And so now we want to see how M18BP1 in presence of ATP will allow this to happen, right? And so this will beautifully explain this at the mechanistic level. And also there is though another question, which is like how MCPH1 that binds in a similar way to M18BP1 has a complete opposite effect, right? So this promotes, you have this binding interface there, and then the outcome is completely different. And this is because we think, we are pretty convinced there are additional domains. So that binding pocket that we have seen, it's an anchor point for these 2 factors. But then there are accessory domains that do completely opposite function. And hopefully we'll be able to clarify this relatively soon. And with that, I really wanna thank the people that did that. This was, the condensing 2 project was originally done by Erin Katz. Now she's a group leader in Sheffield, which originally found this mis-18BP1 in one of her immunoprecipitates. And then Ale really, which you will see here, took over and really in a matter of 2 years really pushed this. He really worked really hard and at times was frustrating and just like this sample doesn't wanna just he tried that, but then ultimately as always there is not such a thing like as a sample that doesn't want to go. It's a matter of trying different things, having a rational approach, and you will eventually get there. And so I have to also, to be honest, the infrastructure, I didn't talk much about the institute, which is brand new. The infrastructure that we have for structural biology is amazing. We have received and we keep receiving quite generous funding, and this would have, what we have done in these 2 projects would have certainly not been possible with just having not regular access to a microscopes and having decent amount of time where you can explore things. Even though data collection are super fast these days, still the luxury to collect, I don't know, 2 million particles data set is not. And there were many failed data collections, which obviously I'm not showing. So, but that's it. So that's the only message I want to reinforce. And I mean, really, thank to the people that made this possible. And thank you.
First question, because then I'll move up.
Yeah.
Amazing work. I'm still trying to process it. I have a question that you didn't allude to, or maybe my understanding is very important. The condensing 2 starts, and creates a loop, and then condensing 1 comes and creates 4 loops. What is creating the loop? Is it just by chance, or are these motor proteins that thread the DNA?
Yeah, I didn't.
Why does condensing 2 only start working on the already excreted loop and not on the straight DNA?
Yeah, the idea is that because condensing 2 is nuclear throughout the entire cell cycle, it starts because it's already there. It's probably one of the immediate events that as soon as you have the CDK1 activation, it starts. Condensing 1 can only enter the nucleus after the breakdown of the nuclear envelope, so it comes later. However, I think there are possibly really differences. So one possibility is really that condensing 1 prefers these pre-made loops by condensing 2. We do not know how these complexes, for example, work together, right? So this is something we want to check on single molecule levels. There are also some other factors which are really essential. I didn't mention there is a chromokinase in KIF4A and topoisomerase. So those 4 factors are really the one that makes these things happen. If you knock out one of those, you always end up with defects. And we now have all 4 of them, and we know that KIF4A, for example, interacts with condensing 1, but how they cooperate, and topoisomerase 2 as well. So the idea is really now to dissect with this mechanistic study what happened. Then the mechanism, it's a loop extrusion, is still not entirely clear. There are a lot of structural arrangements by which you have this topological entrapment of DNA within the globular part of condensing, not so much the ring. But then you need the DNA to be topologically entrapped into this ring. This is more for bringing together, it's important for cohesion, for example, for sister chromatin cohesion, etc. But it's still not settled. It's not settled how this works. So we know that they make huge step because they can, one single step of this, either condensing or cohesion, can process through I think kilobase of DNA, something, but it's still not entirely clear. I think having this reconstituted system, those are the first one in humans. So far, all the work has been done with the yeast one. Will really help us understanding that.
And just to follow up, in your C-trap experiments, you're not seeing the looping yet. You're just seeing the binding.
No, we can't. I mean, we infer looping in single molecule. So there are few groups that have managed to see these extruded loops, but this you have to do it only on beads with a really large amount of intercalators. And I'm not saying that this is an artifact. Absolutely, I think it is how it is, but it's very difficult to visualize it on C-traps because again, it's a confocal, so you don't see that you have to use flux to do it. It's possible that you don't see. Nobody has ever managed to really see a proper loop. I would contend only that also on these glass beads, etc., you do see a structure that could remind you of a loop, but it's not. Yeah, I think like atomic force microscopy, or eventually, a clever way to stop these things in an EM structure looking at. But this is tough. But I think eventually we'll get there. So it's a matter of again work using clever ideas. Maybe once you have structures with DNA, you can think about reactive cysteines that can block maybe that process at specific, the specific step, etc. But I'll see you later. Yeah.
So I got a question on the, let's say, the viral defense model.
Yeah.
What you showed was DNA virus, right? So
Yeah.
I presume this is only working on DNA viruses.
Yeah.
Is there
That's the
anything
idea.
known to RNA viruses that do a similar type of mechanism with the polymerase, or?
Not with RNA polymerase. So it's what elicits the immune response is actually RNA. So the idea, because you have this is called reguy. It's an effect that recognized, again, structured RNAs, uncapped RNAs, and this really activates this receptor, and this gives rise to the interferon response, basically, right? And so the idea is that polymerase not only recognizes this DNA by virus, but transcribes part of it, and it's the RNAs which then elicit it's the RNA transcribed by Pol III that elicits the immune response through a common
anything else.
No. It's just, but of course it begs for the question whether other auxiliary factors are needed, whether some transcription factors are needed, or just recognize the plant double-strand DNA or single-strand DNA. And when does that happen? Viruses are pretty precise in what they do, and because this is the model, right? So a virus enter, and then polymerase recognize the DNA, but the question of when is that DNA exposed to a level also that would elicit a response? And I think, in my opinion, it's more likely that then once the virus has entered into the nucleus, starts to replicate, and then you have budding of the virus out of the nucleus, being, virions being assembled in the ER. Maybe it's there where you have like a couple of nucleic acids, and maybe some of them are not formed correctly, and you might have some leak. So it's not the initial events, but rather, well, also because it's amplified at that point. It's not like one single viral molecule. So that's what we are after. Just try to understand that. There is a question also why only certain type of DNA viruses. We think that maybe it's more widespread than originally thought. Maybe it's just not luck or unluck that those patients were identified only in varicella, because this is like a virus that affects everyone, but maybe there are other less common DNA-based viruses that are also going through that. Showing HSV is one of those. And maybe there are patients also there that have mutations in polymerase, which we don't know yet. Yeah.
I got a more of a general question. Maybe it was really very cool to see. Also cool to see that you also use AlphaFold for as a tool for the whole project. I was wondering, do you always trust the AlphaFold as compared to your cryo-EM pictures, and do you always compare, or is there?
Cases
you really
it's really a valid question, and I think we are at a specific. I think the AlphaFold is truly revolutionary. It is no doubt that this has changed the way I can see it in my lab in a matter of. But there is, the major the people that will take major advantage of this is really the experimental structural biologists, because this can speed up, drive. Some of the structure I showed, it was only possible because we could fit AlphaFold predictions into that. Yeah. So if you have now a possible interaction, you can validate it, check whether AlphaFold comes out. But is it the final answer? No, it's not the final answer. Sometimes you can get predictions that are where the confidence score is very high, and but again, I'm afraid also that the larger community of cell biologists, etc., see these as, well, what do you want to buy in a microscope if you just buy more cluster and just run it on AlphaFold 2 or 3 if they will ever open it but that's not the case. That's not where we are, and I don't think we will ever be there, because, again, first of all, to really understand those models, even if a good model comes out of AlphaFold, you still need a structural biologist to really interpret it, not just to validate it, to see, oh, this is a good model, but to understand what it means. If your end point is to create a beautiful, colorful picture, maybe it's okay. But if you really want to understand what is the biological function and relevance, you still need this. And I think you can't escape to have some sort of validation from biochemistry, from structures, from combination of all of this. Where we also use, I didn't mention, we use quite a lot, this is incredibly powerful, cross-link, so mass spec coupled with cross-link. And when it's done properly, we had the luck to hire a guy that set up like an infrastructure and some sort of facility for us from Yuri Rapsilber lab, and which is, and they use this very precise cross-linker with this STA cross-link. And really, you can get beautiful results in terms of also starting to have architecture really of these complexes. Surely validate your EM structures or other things. And then, so I see this as a combination. It's really like the integrative structural biology approach that if you want to work on really complex problems, will give you the answers. But
Thank you.
Yeah.
I have a question regarding the condensing. Because you mentioned that the Pol III doesn't require energy to bind to, let's say, the particular RNA, right?
Yeah.
So,
well, not to open the DNA.
Sorry.
Yeah.
So it would be there at the centromere at a given time during the cell cycle. So what activates, let's say, the Pol III to start condensing or bringing in the condensing to?
No, this is not the. No, it's not. It doesn't go there. It's not reached there because of Pol III. So the enrichment of centromeres of condensing 2 is probably by this mis-18 BP1 protein which we discovered, and this is unrelated to Pol III, or TF3C. But you have a point, because I showed at the beginning that there is this co-localization between TF3C and condensing complexes. What do they do? So I don't think that TF3C is recruiting condensing, but I think it's possibly a roadblock. So these complexes, they extrude DNA loops, they go on till they reach a certain point where they're blocked. And what it is usually macromolecular complexes or other roadblocks. And I think TF3C is that. And I think it's important because of all these many sites that you have across the genome with retro elements where TF3C can bind, that maybe TF3C has this role of in a way regulating how many loops you form, because you stop there. But this is very speculative. We have to.
Then the active transcription.
No, it's not.
It's not required.
No. And there is active transcription at centromeres. Yeah, this is usually Pol II of the satellite, etc. I don't remember whether there is also some pericentromeric Pol III. No, I don't think so. No, but it's not a transcription active. Sorry, I mean that was a little confusing. Yeah.
Maybe a bit more of a technical question. So
Yeah.
You mentioned at some point million particles, and you're like, well, 1%, but you got 3 or 4 structures, so that's already a big achievement.
Yeah, that's right.
At some point, Alessandro was also working here on time resolve.
Yeah.
Are you doing the same now?
Well, yeah. Well, these transcriptional complexes are they're slow. It's not I mean, for having a full elongation transcription, you incubate it for you have and you check the reaction is 10 minutes. So in that case, I don't think it could be beneficial, yeah, for certain projects if you wanna capture maybe DNA opening or things like that. I think that time resolve that's definitely, for instance, in the condensing work, that might well be, one of those things where you incubate with an ATP analog, and then boom, you give ATP, and bam, and maybe absolutely. So in the case of these transcriptional complexes, the problem really there is a combination of things that go wrong when you vitrify things. Just it's random. Some of them just denaturate, some explode, some whatever. Cross-linking is, you cross-link a lot of things. Sometimes you cross-link too much, sometimes too less. Of course, I mean, it is obviously an artifact that in certain cases can stabilize artificially some intermediates which are maybe very low abundant, but then because in the context of a cross-linker, they are favored because you have 2 lysines which are then all of a sudden closed. You reach for those and all that. So it's a combination. I mean, this is very common. It's not just us. If you look at these big transcriptional complexes, that's the way to solve these things, right? So you collect a lot, and then likely you have, some very, very specific subset that are average well when the majority is. I don't even want to say it's rubbish. It's just maybe you are exploring like a continuum of conformations that exist, and they will never average out because whatever, they are very distinct, etc. So I think in that case, I don't believe that maybe TimeResolve could help and make it better, but I think we are certainly in need of better tools to do it properly, etc., which is going to happen or is happening already. So I think it's very exciting. It reminds me of what happened with crystallography 25 years ago, or maybe a little more, where there were some technical advances that made it possible really to become relatively straightforward. As long as you got crystals, somehow there were methods for doing it in a while. 30 years ago, solving a structure was an accomplishment per se. I mean, and EM is quite rapidly going in that direction. So we still need to have a better way to vitrify samples in a more controlled way, like that. Just like when I have this thickness of ice, not a complete. Sometimes you do the things twice, and because the moon was in a slight different phase, and you get different results. So I think, like, I always encourage when my people get really bad results, it's like, oh, this really look great, and negative staining, anti-cryo, and really shit. Just like, how many times did you try? How many, once. But I screen many grids. Just give it another go. Try maybe something slightly different and see. And if, you try 2 or 3 times with that type of biochemical sample and it's always not good, then okay, once is not sometimes it's really a matter of a different plunger. You tried on a vitro board, go on. So it's a little bit artisanal still, which was how crystallography was 25. Now it's different. I mean, it's also almost dead unless you're working on very small domains or complex with drugs, etc., which is still important. But in my lab, I mean, we could in principle do crystallogenesis, etc., but I don't think anyone is doing it. I mean, we also work with really low abundant mega complex, so there is really no reason why one would do that to themselves. Okay.
If you have any further questions, then let's thank you.
Yeah, let's go for drinks. Thank you, guys. Really enjoyed.